Wonderful. Well, it is my absolute pleasure to get to introduce Aaron. I've known Aaron for a while. I think we first met in Hawaii at the IEEE conference, and not to give a long intro, everyone's here, so they already know how awesome he is. But the one thing I'll say is everything Aaron does is so much fun. It's just like his work is really fun. It's really creative. It's not like anything else anyone else is doing. It's always just like really thought-provoking and refreshing and really great. And the one sort of few points I'll mention is an introduction. He did his PhD with Steve Quake, who many of us know for his work in microfluidics. And then Aaron did something that I think is really, sort of fits his unique work and his creativity. He went the opposite direction that many people go. He went to China to be a postdoc. So he went to Peking University as a Whitaker was it Whitaker fellow or Ford Whitaker?
Yeah, I did both. Yeah,
both.
I had to string along a little bit of funding.
So he did both the Whitaker and the Ford to do his postdoctoral fellowship with Yan Yi Wang at Peking University, and now he's been an assistant professor at Berkeley for 4 years now, I think. Right?
Yeah,
4 years now. And so I had the great pleasure actually to be out to Berkeley for a seminar right when Aaron had just started his lab. So everything was brand new and just had like 1 or 2 students. And so it's been just been so great to see this work progress. And I'm so happy that everyone's come and Aaron can share this with us. So with that, I will turn the floor over to Aaron. Thank you.
Great. Thanks so much for the invitation, Dave. I really appreciate it. I also wish I could be there physically with you all. I was actually planning to come to Philly in the spring to join the single cell meeting. And I had this whole wild itinerary where my wedding was going to be right after. And of course that was canceled, and also my wedding was canceled. So, but hopefully both will be postponed and not canceled. But this is a really great opportunity to share some of my recent work. So I'm in the bioengineering department at UC Berkeley, as Dave said, and I'm also affiliated with the Comp Bio program and the biophysics program. So students in my lab come from all 3 of those departments. And the focus of the lab is to build tools, both software and hardware tools, to study biology on the single cell level. I myself come from a kind of physics and engineering background. And so with each one of these new platforms that we've been developing, it's been really exciting and sometimes intimidating as we voyage into new biological systems. But it's been really rewarding and I think and a lot of fun. And so in this talk, I'm going to talk about two new techniques specifically for generating and then processing multimodal single cell data. And I'm going to start with a software tool that we've developed in collaboration with Nir Yosef for analyzing multimodal single cell data. It's a little bit more mature, and the experimental techniques are commercial. And then I'll transition into a new microfluidic platform for harvesting a new kind of multimodal single cell data, and we'll kind of explore what it's good for, or what it may be good for. Okay. So to start, I think, since we've been able to detect single cells with microscopy, with every new technology to harvest information from single cells, we found out that there's more and more types, or unique identities of cells. And there's many different axes with which we can quantify this cellular identity. Of course, oftentimes we define cells by where they come from in the body. Most cells in an organism have more or less the same DNA, but we can quantify the unique identity of a cell by its copy number variation or single nucleotide polymorphisms. A cell's identity might be based in part on how that DNA is organized in the nucleus, which then informs the genes that are expressed. And I think over the past 10 years, single cell RNA sequencing has really emerged as a powerful, high throughput and quantitative way of profiling the identity of single cells through quantitative measurements of gene expression, unbiased quantitative measurements of gene expression across the whole transcriptome. Of course, for a very long time, people have been characterizing cells by the proteins or metabolites, so functional molecules that the cells make. Um, and and really, microscopy is still one of the most powerful, and it's certainly the oldest single cell measurement. It's the way that we knew, looking at cells and seeing what they look like was the way that we knew that there were in fact single cells. And these are different ways these are different properties that we can measure. But there's also, a lot of different ways that we can think about cellular identity. We can think about what state in a developmental trajectory that cell is in, what lineage it came from. Is it a disease state or is it a healthy cell? What's the function? And in this way, single cells are a lot like humans. Even the same type of human, even humans with the same job, like professors, can have a vast array of identities. And for any given person, we can kind of identify them by asking similar questions. What's your genotype? Where are you from? What's your family history? What's your job? How much money do you make? Are you sick? How old are you? And in this sort of analogy, I want to make the argument that just like when identifying, let's say, the population of a society, it's really important to be able to make measurements on the single human or single cell level. Because in a country, it might be interesting to probe what the average income is. But a more important measurement is how that wealth is distributed. Because two countries might have similar average incomes across the whole population, but very different distributions of wealth. And in the same way, as we make single cell measurements, it allows us to create, much more accurate distributions of the heterogeneity of cells in a given system. And I'll extend that analogy even further to say that it's not enough to make individual measurements of any one of these axes of identity to quantify what's going on in a complex biological system. For example, when we take the census, it wouldn't be very useful if we sent out 100 different censuses and collected one bit of information from the whole population on each census. For example, we get one that tells us the distribution of age, another decoupled measurement of the distribution of healthcare, or another decoupled measurement of somebody's, of the workforce or the educational attainment. It's really important for a census to have one questionnaire that has all the questions, so that we can make these coupled measurements and therefore infer relationships between different aspects of our identity. So how much do people make who have this level of education attainment and have this genotype and live in this area, and have access to this kind of healthcare? And in this way, by making multimodal measurements, we can learn not just about the nature of the distributions of these individual metrics, but actually learn how the interplay between these. And so in the same way, we've been trying to develop tools that allow us to make measurements of these different aspects of cellular identity in the same cell, so that we can not only map out the distributions, but see, for example, how chromatin organization informs gene expression, or how morphology has a sort of circular feedback with the proteins and metabolites that are produced in that cell. So with that, my lab kind of focuses on 3 separate technologies that we integrate together to make these multimodal measurements: microfluidics, microscopy, and genomics. And by genomics, I mean sort of turning aspects of a cell into DNA so that we can sequence it on a next generation sequencing platform and harvest that information. And a classic example of that is single cell RNA sequencing. And then more recently, we've been collaborating with some colleagues in electrical engineering and computer science to figure out, now if we have a lot of different data from many different single cells, how can we unify that really high dimensional data into a single low dimensional representation, so that we can understand how cells are distributed across this multifaceted identity space. And so we've been kind of, foyering into a little bit of computational pipeline construction as well, so that we can start to gain biological insight from these measurements. So going back to this paradigm, just a quick intro before I dive into the specific projects with some of the work that we've been doing in the lab. We've been working on a microfluidic platform that can allow us to couple stimulated Raman scattering images of single adipocytes with gene expression. And so for every cell, we'll get, for example, this distribution of lipid droplets that might indicate the stage or the type of adipocyte. And when we also get a high dimensional gene expression data set, and both of these identities can be classified in their respective space. And so we've been trying to think about how can we then unify the different parameters that we can harvest from imaging data with the thousands of parameters that we can get from gene expression data, so that we can get like one sort of identity of that cell and see how different cells are different in a unified space. And then in collaboration with Yang Huang, my postdoc advisor, we've also been using stimulated Raman scattering to image tissues on the tissue level. And then, so, by using stimulated Raman scattering, we can avoid fixation and staining of tissue, and we can recapitulate some of the morphology that you would get with like classic H&E stains. And you can identify different regions. This is a tissue section from oral squamous cell carcinoma. And then we can go back in with the same laser that we use to do stimulated Raman scattering and excise a micro tissue that we can identify using the stimulated Raman scattering image, then recover that micro tissue and perform DNA and RNA sequencing and look at the differences of molecular composition as we go through different places of for example, a tumor section. So in this first main story, I want to talk to you about a really nice collaboration with Nir Yosef in the EECS department here at Berkeley, where we've been using a fairly mature microfluidic technology to probe whole transcriptome landscapes of single cells and the surface proteome of single cells in the same cell, but really working on figuring out how to merge those two data sets so that we can analyze not only individually gene expression and surface protein expression, but the interplay between those two aspects of cells. And so we've been using a technique called CiteSeq, which has been developed by Rahul Satija and Peter Smybert in the New York Genome Center. It's a very powerful technology that's been around now for maybe just over 5 years or so. And at the risk of a little bit of redundancy, I'm just going to go through how this technology works for those of you that don't do single cell microfluidics. And the idea here is it really, CiteSeq piggybacks off of the coupling of two technologies that have really kind of pushed the single cell gene expression measurement into a high throughput realm so that we can start to characterize thousands to tens of thousands, even hundreds of thousands of cells at a time. And one of the core units of this technology is the barcoded bead, right? So this is a solid phase or hydrogel bead that's colored with these single-stranded DNA oligonucleotide barcodes with a very specific sequence that identifies which bead that barcode is from, using what's called a cell barcode. So all of the oligos on this bead have the same cell barcode. They have a PCR primer so that we can amplify whatever captured by this barcode. And then they'll have a poly-T tail, which can be used to capture the polyadenylated tail of mRNA and filter out all the non-polyadenylated mRNA in a cell. And so now when you perform, if you can couple some RNA with this bead, if you can couple this RNA from a single cell with this bead, and you perform reverse transcription, then you can imprint the barcode from this bead onto that complementary DNA. And every one of these barcodes also has a random unique molecular identifier so that when you amplify using PCR all of these cDNAs, you know exactly not only which cell did that RNA come from, but which template molecule did that amplicon come from. So then you can sequence the cDNA library and you can count all the transcripts from all the different genes and all the different cells. And so what CiteSeq does is it adds a barcoded antibody to this workflow. And so the idea is that instead of like a typical FACS pipeline where you'll attach a fluorescent a molecule to the antibody of interest that probes the protein of interest on your cells, you start with a pool of antibodies that each have a unique oligonucleotide barcode that's conjugated to the antibody. And so each barcode is specific for which protein that the antibody is designed against. And then each one of these barcodes also has a poly-A tail, so that if you stain your cells with this panel of barcodes, with barcoded antibodies, and wash away all the ones that don't bind, and then run a regular drop-seq sort of platform, And in addition to capturing the RNA, you also capture the abundance of surface proteins as labeled by these barcoded antibodies. And so to get this technology to work, what you need is you need a suspension of uniquely barcoded beads. You need a pool of barcoded antibodies that you can stain your cells with. And then you need to mix these together in a microfluidic droplet generator. And fortunately, this represents, maybe 2 decades of technology development from people all over the world. And nowadays you can buy both of these things commercially. So BioLegend supplies barcoded antibodies, and 10x is an example of a company that makes these droplet generators for single-cell RNA sequencing. And the idea is that you can put your beads, your cells, and your reagents into the inlet, and then in 2 other inlets you also put oil, and you can generate droplets at a high throughput level. Many of you do this regularly in your labs. And then so you're able to generate thousands to hundreds of thousands of droplets in a single experiment. And if you tune the concentration of beads and cells just right, then it'll be a rare chance that more than one cell or more than one bead will be in any droplet. And so if for example 1 out of every 10 droplets has a cell, and 1 out of every 10 droplets has a bead, and 1 out of every 100 droplets will have both. So you can couple a single cell with that unique barcode, and then you can perform your reverse transcription and amplification in these droplets. So this is a pretty common technology. 10x actually has figured out a way through some innovation from the David Waite's lab and Adam Abate's lab at UCSF of loading every single droplet with a bead so that you can have higher throughput. But the result of this is a lot of data, especially if you have a lot of barcoded antibodies. So in this experiment that I'm going to talk to you about, we've had a couple hundred barcoded antibodies. And so the result is that you have thousands of cells, each with a matrix of with quantities of thousands of genes, and then also hundreds of proteins. And the question that a lot of people have when they process this kind of data is, okay, well, what do we do with this? Because you can't look at this data and interpret it. There's too many variables to visualize the different populations of cells that you might have. And so what people have been doing for a long time now is that they reduce the dimensionality of their data set into a lower dimensional space. And so the classic, right now, a standard approach is to reduce your high dimensional data down into 2 dimensions using principal component analysis or some other nonlinear dimensional reduction. And what we might do with this multimodal data set is we might plot the 2 major principal components of all the gene expression into 2 dimensions, and then map the level of a given surface protein onto that 2-dimensional space. So for example, if maybe there's 2 clusters of cells where each dot in this schematic is a cell, and cells that have similar gene expression profiles are clustered near each other. And then you might map, using a color bar, the level of the surface of the abundance of a specific surface protein, like here I'm saying CDX. And so you would see that, oh, this surface protein marks this cluster, this other surface protein marks this other cluster. And you might have surface proteins that are actually ubiquitous on both clusters, but have gradients of expression levels. So this is one really interesting way of sort of harmonizing gene expression data with surface protein abundance. The problem is that if you start to have hundreds of proteins in your panel, it becomes, at the very least, labor intensive, and at the very worst, kind of impossible to really find patterns between gene expression and the network of surface protein abundance. What do you do if you have 200 proteins? And so in collaboration with Nir Yosef and 2 joint graduate students from the bioengineering department and the computational biology department, Adam and Zoe, we developed a computational framework for analyzing this sort of paired kind of data. And it's built off of SCVI, a variational autoencoder that Nir and his lab developed over the past few years. It's basically a neural network that can take all of the data from your multimodal, your site-seq experiment, and then generate a reduced dimensional space so that we can perform downstream sort of analysis that's classic to single-cell RNA-seq, like visualization, clustering, differential expression, and so forth. And so what we did to test out this new framework that we're calling total VI, or total variational inference, is we ran an experiment with 2 mice where we would have a fairly controlled system that with cell types that are relatively well characterized, so that we can benchmark the performance of total VI in this high dimensional data set. And so some of the challenges that you run into when you're doing single cell RNA-seq or other kinds of single cell measurements is that A, these measurements are really noisy, and B, these measurements are very susceptible to different sort of systematic experimental variation. For example, the differences between mice, or the differences between 10x runs, differences in experimental batches can yield differences in your measurement. And so we designed an experiment where we harvested the spleen and the lymph node from two different mice, and then we split those single cells up into to 8 suspensions actually, and we incubated one suspension with a panel of 100 proteins or 100 antibodies, and we incubated the other suspension with a panel of 200 antibodies. And so now we're able to kind of probe the ability for total VI to deal with these experimental variables. So mouse, day, which day we sequence, sequencing run, and also see how we can handle harmonizing data that comes from different kinds of measurements, different protein measurements. So what we get after we do this experiment is we get a large, I represented this a moment ago as 2 different matrices, but you can imagine concatenating these matrices. And so what total VI does is the variational autoencoder this is based on a deep generative model, which essentially learns a reduced dimensional latent space or hidden variable space that's about 20 dimensions instead of the 1,000 by 1,000 dimension of the original data set. And the idea here is kind of a lot like image compression. So the notion is that the identity of a cell is not necessarily as complex, doesn't necessarily require a 10,000 vector space to identify. That because of co-regulated gene expression and protein expression between cells of similar types or similar states, that there's a lot of redundancy in this data, in the same way that there's high correlation between pixels in an image. You can compress this data into a reduced dimensional space. And so what total VI does is it uses a model that we define up front. So we use a negative binomial distribution to model the distribution of RNA counts. And we use a mixture of 2 negative binomials to model the distribution of protein counts. And total VI learns the 20 dimensions that can recompute the distribution for each one of these variables. And in every round of training, it updates the value of each dimension based not only on the estimate of expression from the measurement of that gene in that cell, but also based on all of the measurements of all the other genes and all the other proteins in all the other cells. And so it learns a probabilistic distribution of the likelihood that any gene or protein is expressed at a specific level in any cell. And then it recomputes, it decodes this latent space into a new full dimensional matrix that has a smooth and denoised probabilistic distribution, so like a likelihood estimate for every variable, so that we can perform statistical tests and look at the chance that there's a differential expression between this gene and that gene, between these cells and those cells. And so in this latent space, we can also do stuff like batch correction and denoising, and I'll show you what I mean by that. So if you were to just concatenate the surface protein abundance matrix with the RNA expression matrix, and just do a linear principal deconvolution with principal components, and then plotted all your cells from these 2 mice, and this is the case with the lymph nodes specifically from the 2 mice, into this 2-dimensional reduced dimensional space, then you would see different populations of cells. So the B cells kind of cluster together and the T cells kind of cluster together. But you also see that the cells from the different mice also cluster together. So in yellow, we're looking at mouse 2, and in blue, we're looking at mouse 1. And there, because of differences between the mice, then those differences are reflected in the lower reduced dimensional space. But when we harmonize the data in the latent space of total VI, we're able to correct, because we know which cell came from. Total VI can correct for differences between the 2 mice and remove that from the latent space. And therefore, when we use the neural network to do dimensional reduction instead of the linear deconvolution, we can remove that batch effect, and cells of the same type, even though they're from different mice, will cluster together in that reduced dimensional space. That's one of the technical artifacts that Total VI is able to deal with. And there's many normalization techniques that can also remove batch effects. The neural net just does a really good job of doing that in the same space. And so, if you have different numbers of proteins or different numbers of cells, you can still deal with all the different batch effects from experiment to experiment. And in fact, Total VI can even impute the values of proteins that you don't have in one of your batches. And so we were able to show that the cells that only have 100 proteins were able to still overlap with the cells in which we measured 200 proteins. Another really important thing about this kind of data is that with single cell RNA-seq data, it's pretty well known that it's a very sparse data set. So, there's not a lot of molecules. I mean, there's a couple 100,000 maybe RNA transcripts in a single cell. And when we try to capture and sequence those molecules, there's a very low capture rate. On average, we're capturing around 5 to 20% of the transcripts in the cell. And so the gene expression matrix for single cells is typically a fairly sparse matrix. This is not real data. This is just like a toy data set. But you see there's a lot of zeros. The protein data is actually quite different because of non-specific binding, not just non-specific antibody binding, but when you incubate your cells, you have 100 different antibodies. And depending on how well you wash, or depending on whether or not antibodies are sticking to each other or sticking to their DNA barcode, or depending on whether or not barcodes fall off the antibodies, you end up with a lot of background. And when you're doing FACS, you can easily calibrate for this background by just setting your gain or setting your zero at the level of the background. But when you're doing this sequencing measurement, it turns out that even cells that don't have that surface protein have a finite measurement. And so for this reason, we use this double negative binomial to model both the background signal in the protein measurement and the foreground signal. And so, for example, like on the top histogram, I'm showing the histogram of RNA counts for, let's say, for the transcript that codes CD8. And you can see that there's a lot of cells that have 0 counts, and then a lot of cells that have some finite log count. And in the protein measurement, you have a lot of cells that have a background that aren't expressing CD8 on the surface, and then a very clear population of cells that are expressing CD8 on the surface. And now this is really easy to separate the foreground from the background. If you were doing FACS, you would just draw a gate right in the middle, and you wouldn't even have to think about this background. But what Total VI does is it, in addition to estimating the likelihood of estimating the abundance of a particular protein, because it is computing, because it's learning a probabilistic distribution for every variable, it can also give you a likelihood that that signal is actually coming from the foreground or the background, because it compares the value for that cell to the value for cells around it. And it says, okay, you might have had a lot of counts for this protein in this cell, but all the cells around it had a low count. So this was probably a background signal. And so here I'm plotting in the scatter plot for every cell. On the x-axis, I'm plotting the log count of that surface protein, CD8. But on the y-axis, I'm plotting the probability that that measurement is real. And so you can see that indeed for CD8, because it was so easy to separate foreground from background, that the cells that had a high protein count were probably real, and the cells that had a low protein count were probably background. And indeed, we can look at these cells in our UMAP space, and the cells that had CD8, were the CD8 T cells and the B cells, and the cells that didn't cells that had CD8 were the CD8 T cells, and the cells that didn't were the CD4 T cells and the B cells. But now, what if you look at a histogram for a protein like CD28? This was an antibody that maybe wasn't titrated so well. There was a lot of maybe non-specific binding. And so you could still draw a gate here, but you wouldn't really know where to draw that gate. Some of these cells have real foreground signal, and a lot of these have background signal. But now with Total VI, when you look at the probability that that measurement is indeed foreground or background, you can draw your gates in the probability space. And so now you can look at CD8, which is a marker for T cells, and you can say, both these cells that had a low count but a high probability foreground were indeed all the T cells, whereas the cells that had a low count or sorry, the cells that had a high count but a low probability of foreground were some of these background were just noise in the B cell population. This is recapitulated in the RNA data. You can see that the T cells had a bunch of, in the histogram, had a distribution of cells that had high RNA count, and indeed the B cells had no gene expression of RNA of CD8. And so in that way, we can total VI can actually separate foreground and background and create from this otherwise mixed distribution a very denoise distribution where the new values represent only the real values with high probability foreground. And so with this kind of data, you can annotate your clusters, which is a sort of a typical step in the workflow for a lot of these human cell atlas projects. You can cluster your cells in a combined gene expression, protein expression space. And then you can go through and start to identify which cluster is which based on not only the genes that are being expressed in those clusters, but also the proteins that are being expressed in those clusters. And so here, for example, I'm showing that CD93 is an early B cell marker. You can see that lights up both in protein and in gene expression in the transitional B cells. So this is still actually a manual process. Zoe went through and literally looked at each cluster and looked for specific protein markers that were expressed on the surface, but also genes that were expressed externally that maybe didn't have a surface protein. And she was able to annotate these clusters by hand using the combined data set. So, all right, how are we doing with time here? Let's see.
It's 2:33.
Great, perfect. So we've been using this platform, we've embarked on this very cool collaboration, again, with Nir Yosef and Ellen Roby, an immunologist here at UC Berkeley, to look at the development of CD4 and CD8 single positive T cells in the mouse thymus. And so what Zoe's done is she's taken two knockout mice and two transgenic mice that have been genetically engineered to only produce either CD4 single positive T cells or CD8 single positive T cells in their thymus. And then she's also collected a whole bunch of cells from a wild type mouse that has both CD4 and CD8 single positive cells in like a normal mouse. And she's able to map all of these positively selected T cells from these three mice into this same reduced dimensional space using both the protein and the gene expression data. And so here's sort of a graphical representation of that bifurcation. So over on the left here, again, each dot is a cell, and in these two dimensions are, the reduced from the 20-dimensional latent space of total VI. And so you can see this left lobe over here are the CD8, CD4 double positive cells that are high in both protein and RNA for CD8 and CD4. Although you can see that the RNA measurement is sometimes sparse for CD4. And so you can recover a lot more fidelity with the cell types using the combined measurement. And then you can see as these cells develop, and these are all at any moment in the mouse thymus, when the mouse is 2 to 4 weeks old, it contains all stages of differentiation of these two cells. And so with one biopsy, we can get sort of the full spectrum of developmental trajectory in both CD4 and CD8 single positive. And you can see this development really well. So this is the CD4 branch right here. You can see CD4 protein lighting up. You can see the CD4 transcript lighting up, and you also can see ZBTB7B, a zinc finger transcription factor that regulates commitment to the CD4 lineage. This is something that you obviously couldn't measure with surface proteins. And and equally, you can see the trajectory of CD8 single positive from double positive all the way to the mature single positive CD8 cells. And here again is RUNX3, another regulator marker for the commitment to the CD8 lineage. So now that we have this sort of trajectory through developmental space of these two cell types, we can line these cells up in time based on whether or not they went down the CD4 or the CD8 lineage, and we can assign a sort of a pseudo. This is a common procedure now in single-cell RNA-seq data. We can assign a pseudotime address for every cell that's sort of mapping the probability that that cell is at a given stage of its trajectory. And then we can plot, then we can plot not only gene expression as a function of developmental stage, but also surface protein abundance as a function of developmental stage. And the key here is we're not just now annotating, different kinds of cells in this heterogeneous population, but we can actually begin to learn about the relationship between the expression of that transcript and the presentation of that surface protein on those cells in the same cell. And so what I'm plotting here is I'm lining up cells in time, and I'm plotting a smooth expression in blue of the transcript for a given gene. And then in red, I'm plotting this abundance of the surface protein for that same gene. And you can see, for example, in CD5, which is an early was a T cell marker, you have this spike of expression of the transcript, but then the protein itself lingers and stays on the surface for a lot longer. CD69, which is a marker for early activation of T cells, it spikes in the transcript, and then it spikes in the surface protein, but then dies off as the cells become mature. And then if you look specifically in the CD4 trajectory, for example, you can see the decrease of expression of the CD8 transcript, and then a lagged removal or clearance of the CD8 protein from the surface of that cell. And so in this way, you can begin to infer not just what's like cell types or cell states, but you can learn about some of the processes that underlie the transition of these cell states, and maybe even the kinetics of not only protein translation, but also trafficking to the surface of the cell. And so Zoe really has some really nice work where she's able to then go back in time and look at, pull up this slide right here. So go back in time and look at gene expression and protein expression patterns that are upstream of some of the branching point, to learn about which transcription factors and what signaling pathways can predict which direction any given cell is going to go. And that's a project that's ongoing with Ellen Roby right now. Okay, now I'm going to switch gears to a new platform. So maybe should I take questions if anybody had any questions about the site-seq data, or should I save that to the end?
Why don't we save it till the end? That way we can just do, unless anyone has any burning questions that they want to ask right now. I think we're good. Let's just keep going.
Great. Okay, so that technology is fairly mature because you can buy a 10x platform and you can buy the antibodies from BioLegend, and then you can run total VI. And there's a manuscript on the bioRxiv. You can see all the different benchmarking measurements of total VI. This next platform is a little bit more experimental, and it addresses how we might learn about chromatin organization in single cells using two different measurements. So two sort of canonical ways that we can understand how chromatin is packed into the nucleus of a cell, either in terms of its three-dimensional organization or just in terms of the binding landscape of a particular DNA binding protein. We can image or we can sequence. For example, we can do ChIP-seq, and we can pull down fragments of DNA that are bound to proteins of interest. We can sequence those fragments and we can see where the protein of interest is bound to the genome. Or we can label a protein of interest or a histone modification or even certain specific loci of DNA with a fluorescent marker and image where those loci are physically located or spatially located in the nucleus, or image where those proteins are bound to those loci in the nucleus. And so this is a project that's been spearheaded by Nick Altimos, my first PhD student, and Andy Masalan, in which they tried to see if we could unify both of these measurements to see where protein is binding in the genome and where it's binding in the nucleus spatially. And so, we can sort of identify or characterize chromatin organization by looking at where proteins are localized, because we know that there's proteins that localize to specific compartments of the genome in the nucleus. For example, there's chromatin compartments A and B. Those are oftentimes measured with high-C measurements. We can look at loci that are interacting with nuclear pore complex. We can identify the nucleolus and transcriptional clustering or the nuclear lamina. And each one of these nuclear landmarks can be characterized or can be identified by their relevant proteins that are associated with those landmarks. So there's laminar proteins, and there's nuclear pore complex proteins, and there's nucleoli proteins. And so if we can see where those proteins are binding to the genome, then we can see which loci are physically related to these different landmarks, which genomic loci are physically related to these different landmarks in the nucleus. And so a technique that we've been using to probe this sort of just broad question of how do we measure where these interactions are happening is a technique called DAM-ID. And we've been using DAM-ID combined with imaging to answer both of these questions. Where in the genome is a protein binding, and where spatially in the nucleus is a protein binding. So the way DAM-ID works is you choose a protein of interest and you fuse it to DNA adenine methyltransferase, which is a bacterial methyltransferase that will methylate the tetranucleotide sequence GATC, and it'll methylate the adenine in that sequence. And so by creating this fusion complex, you can essentially leave a permanent mark wherever your protein of interest went in the genome. And this mark is orthogonal because there's not much methyladenines in the mammalian genome, and it's not particularly toxic either. I mean, if you were to methylate every single adenine in the mammalian genome, I'm sure something would happen, not good, but at this level, it doesn't have any noticeable biological effects. And so what you can do is now after you've marked the genome where your protein is bound, you can specifically digest the genome using DPN1. You can digest it because DPN1 recognition site is methyladenine and it's specifically in GATC sequences. You can ligate on an adapter, and then you can amplify these regions using PCR. And this is a powerful technique that was developed by Bas van Steensel about 20 years ago, around the same time as ChIP-seq. But what's unique about it is because you can amplify your signal with PCR, you can avoid some of the loss in sensitivity that you have with antibody pull down in ChIP-seq. So you can get kind of single cell resolution. So one of the nuclear landmarks that's been regularly profiled with DAM-ID over the past 20 years is genomic loci that interact with the nuclear lamina. And so by fusing DAM to lamin B1, then any place in the genome that butts up against the inside of the nuclear envelope gets marked, and you can therefore, using sequencing, identify these lamina-associated domains. And it turns out lamina-associated domains are a very important structural scaffold for the genome. It also have regulatory implications. And so, there's over 1,000 lamina, like annotated lamina-associated domains in the human genome. About 30% of the genome is associated with the lamina. These regions are AT rich, and they tend to be gene poor. They're fairly broad regions, and they come in kind of different classes. There are some regions of the genome that are constitutively associated with the lamina across cells and across cell types. There are some regions of the genome which are constitutively not associated with the lamina. Those are oftentimes referred to as constitutive inter-LADs. And then there's regions of the genome that can kind of move to and from the lamina depending on the cell state and depending on the cell type. And it turns out that these regions tend to be gene rich. And sometimes it's been in some really cool experiments. If you dock a particular loci to the lamina, you can generate repression of that gene. And you can also see that certain domains have moved to and from the lamina as cells differentiate into mature cell types, because those genes are either used or not used in those cell types. So in addition to sequencing these loci, you can also image these loci by using a modified fluorescent probe that's a modified DPN1, where its endonuclease activity has been removed and instead it's been labeled with a GFP. And so now, before we perform the DAM-ID sequencing measurement, we can recruit a fluorescent molecule to the location of this protein DNA interaction by recruiting this M tracer molecule to the methylated genome. And as a result, we can see not only where that protein is bound, but we can see where that protein was bound, because this is a mark that's now on the DNA. And so using imaging, we can track where these loci move to after they've come into contact with the lamina. So what Nick did is he developed a microfluidic device that would allow us to trap single cells, image their lamina-associated domains using fluorescent imaging, and then sequence those lamina-associated domains using DAM-ID sequencing. And so in this technology, we use classic microfluidic valves and pumps to to direct single cells to this trapping chamber where we can image them and then send them through a series of chambers to perform the DAM-ID measurement. And so this is how one of those pipelines in this platform looks. You can trap the cell, you can image it with bright field imaging, you can image it with fluorescent imaging and see the lamina-associated domains. And I should say that this was a proof of principle study, so we chose to look at lamina binding because these LADs are very well annotated in the human genome, and we know where they are. They are at the lamina. And so we kind of knew what to expect, and we wanted to test the performance of this microfluidic DAM-ID platform. After imaging the cells, you can recover the enriched regions and sequence. And so for every cell, you get a microscopy map of where the genome has come into contact with the lamina. And then you also get a genomic map of where in the linear genome, physically which loci were the ones that come into contact with the lamina. So here we're mapping DAM-ID maps of single cells across chromosome 1. Here we're also plotting gene density so that you could see that, and then here we're plotting lamina association in a bulk DAM-ID measurement of hex cells. And so you can see, for example, that in this region of chromosome 1, this region was highly associated with the lamina and is fairly gene poor. And then here are traces of single cells that correlate pretty well with our bulk measurement. And here's the correlation scatter plot. And then we came up with a classifier to say, okay, well, what are the chances, because this is also a very noisy measurement, as you can imagine. And so using annotated constitutive LADs and constitutive inter-LADs, Nick developed a classifier to call LADs in single cells. And so now for each cell, we have sort of its laminar spatial map and its laminar genomic map. And if we only express DAM without fusing it to the lamin, then it methylates the genome everywhere. So this is what that kind of cell looks like. And those cells, their laminar, their DAM-ID mapping sort of correlate with each other because the methylation mark occurs throughout the genome. And for cells where DAM was fused with lamin B1, you have high correlation in their lamina association. And then you have some cells that did have the DAM fusion, but presented in the microscopy image a fairly highly methylated genome. And indeed, that cell correlated kind of in between. It was clustered both with cells that had just DAM and both with cells that weren't expressing DAM. So then what we did is we said, okay, well, so for any given loci, what are the chances that that locus is constitutively associated with the lamina? Or what are some regions where from cell to cell, since we have a single cell measurement, we can see regions that are variably associated with the lamina on the single cell level. So for example, all of these regions here are constitutively not represented in our sequencing data. And so we have very low abundance of reads that map to this region. Here we have constitutive lamina association, and this is validated by cross-referencing with a panel of different cell types, all of which constitutively represent these loci to lamina. But then there's regions where for half of the cells, we did get, enrichment, and for half of the cells, we didn't. And so we identified these regions as variably VLADs or variable LADs because they were variable from cell to cell. And indeed, cells that, regions that had variable lamina association tended to have higher average gene expression in bulk gene expression than the regions that were constitutively represented at the lamina. And it turns out that between different cell types, you have different variable lamina associated domains. And so this kind of indicates that there's some regulatory function in lamina association, even on the cell type to cell type level. And this is something that we can probe with microDAM-ID. And so one of the powers of microDAM-ID is we can sort based on very complex phenotypes. And so Nick is, and this is total future directions, but Nick is now using this platform to explore laminar disruption. And so in particular, when chromosomes are improperly segregated during mitosis, you can get the formation of these micronuclei with little bubbles of the nucleus that kind of shed off of the actual nucleus during cell division. And we can induce micronuclei with drug treatments and you can see them. This is here we're staining the DNA with host stain, And you can see these little micronuclei. These arrows aren't really fairly well registered, but you can see these little micronuclei here. And so the idea is we can now sort cells based off of the generation of these micronuclei, and then do DAM-ID to see which chromosomes are variably associated with the lamina in cells that have micronuclei. That's just ongoing work. And some of the other future work is we want to then use this platform to probe not only laminar association, but also transcription factor binding or histone modifications. And it turns out you can also probe chromatin accessibility, because if you just express DAM by itself, it'll preferentially methylate GATCs in regions of the chromatin that are open. So it turns out, and Yapkin has shown this quite extensively in his lab, that you can actually probe chromatin accessibility with DAM-ID. So with that, I'd like to thank you for your attention. This is my lab. These are some fantastic collaborators. The work I talked about in this talk was collaboration with Nir Yosef and Ellen Roby. And these are my students, and I'll take any questions.
Now, first is thank the speaker for an incredible talk.
Thanks, Dave.
And so maybe what we can do is just, let's see, how do we do this? Oh, Jun Young raised his hand. Okay, I can see it. Good. Jun Young, please go ahead.
Great, Aaron. Wonderful talk. I had some question about the integrated analysis. So I guess for site-seq and sort of surface protein, the relationship between the transcriptome and the surface protein, I'm not exactly sure what it should be. but given the autoencoder scheme, let's say you apply this to like ATAC-seq and transcriptome or something like that. From the latent space, do you learn relationship between those two data modes? Besides all the registration and all the automatic things going on, one would hope that you learn something between the the molecular modalities and their relationship.
Yeah, so that's a really good question, and that's what I was kind of trying to get at, because we haven't learned anything yet. But I think that when you have so there's two parts of that question. First, I think it's really important for any of these multimodal analyses to do two things. One, you have to treat each measurement in a bespoke way, because each measurement has its unique noise source and systematic variation. ATAC-seq is particularly sparse. So before you can combine two measurements into the same joint latent space, you need to kind of adjust each one for its different kind of noise sources. That's what we did with the proteins in the negative binomial. And then I think the advantage of harmonizing both data sets into the same latent space is that you can then, in theory, look for direct correlations between features that are not necessarily even the same protein in the same, like in the case with site-seq, I'll just say. You don't have to even look at the same gene and the same protein. You can look at correlations between surface protein and other genes, or surface proteins and other surface proteins. And you can kind of do that in an unbiased way. And so here I'm just plotting across the transitional B cells and the mature B cells, a correlation map of both genes and proteins. And you can see that they cluster based, like within the cluster of B cells, you can kind of, we haven't done this, but you can imagine inferring regulatory networks. But what's really important about this is that these correlations will be completely skewed by the relative noise. And so that's why I wanted to make this point that you really have to deal with that noise independently first, so that the correlations are quantitative. And to your question about, or to your comment about the relationship between the transcript and the surface protein, there's a lot of steps, right? First of all, we may or may not even capture the transcript. So there's noise in that measurement. And then there's translation, and there's splicing, and then there's trafficking, and then there's degradation. And so, I don't know exactly what we can infer, but one of the things I'd love to try is, okay, well, now if we can kind of put a real time into this trajectory axis, then I wonder if we can use some like basic models of transcription, splicing, translation, degradation, and trafficking to, infer some of the regulatory processes between the generation of the transcript and the generation of the surface protein. So we haven't done that yet, but I think it's a really cool question and idea.
Okay, so we have a question on chat from Hannah Hornig. Says, thank you for your talks, Dr. Streets. Does a total IVI probabilistic model make any statistical assumptions when completing harmonization? If so, what are they?
Yeah, so the first assumption that we're making is that each variable is independent. So we're assuming that there's not any, we can find correlation, but we're assuming that there's not any dependence up front between the proteins and the RNA, and we're just modeling them as independent variables. And then we're also the main assumption is that the RNA counts follow. I don't have a slide for this. That the RNA counts follow a negative binomial distribution, and the big shift from total VI from NIR's original SCVI was that the protein measurements follow a mixture of a double negative binomial, because we want to also be able to model that background signal that I was talking about. And so in that way, we're able to then remove that second binomial from the protein. And then there's a lot of other priors that we use in the model itself, but I think those are the main ones conceptually.
I'm curious about the impact of those, sort of prior assumptions, right? So you start with this idea of you're able
were you one of our reviewers? That was the exact exact question. What is the impact of your priors on the performance of the model? That was.
Oh, it wasn't me, I promise. But yeah, I guess it's neat that you're able to take this distribution of the proteins that you see, and then sort of replot that as like how confident you are in each of those measurements. But what about, if the reality is that there are these outlier cells? What if there are these like jackpot weird cells that are doing something really different than every other cell? Wouldn't your model just sort of assume that it's a bad measurement and throw them away?
Yeah, no. That's a really good question, and this is particularly true. For example, let's say you've done a site-seq experiment with 50 proteins, and then you do another site-seq experiment with 200 proteins. In the joint latent space, you're essentially going to start predicting the surface expression of the proteins that you didn't even measure. Like if you want to harmonize these two data sets together, you're going to have to impute or predict the value of the proteins on the cells for which you didn't measure the protein. That sounds cool, but it's potentially scary because it's not real data. I mean, It's probabilistic data. And so you're right. So if you have a population that doesn't cluster, so in a way you can imagine total VI like takes advantage of the fact that cells look similar to make these probabilistic assumptions. And so if you have this outlier population, does it say that it's just not real? And it really has to do with how many cells there are and how distinct that population is, because it models the noise as well. So we have an idea for what the noise looks like. But if you have decent mixing, so basically, to answer your question, we haven't applied this to all the different cell types. And so we hope that the idea is that, in the Human Cell Atlas, people will start integrating these site-seq data sets and learn about, cell types that are kind of unique or don't cluster in the latent space. And in terms of the stability of the model to these different priors, like we did some tests with ground truths that we know, basically with cell systems that have been previously characterized. Like that's one of the purposes of using this this immune cell model, because like people know the different kinds of cells for the most part that are in the mouse thymus, but there's not really a ground truth. So we don't really know when we're missing a cell type that's real or not. But I think this is a common problem in like these larger Human Cell Atlas projects, because sometimes people mistake doublets for new cell types, or they miss new cell types because they thought they were doublets or different things like that. And I think that's, that question might require like bespoke analysis or sort of manual validation for any. I think of this as more like hypothesis generating, not hypothesis validating. That's a really good question.
Let's see. Does the audience have any additional questions? I imagine you must. Oh, cool. See.
And I think we do have a meeting with the students. So if a student, here we have a raised hand.
Oh, we do. Okay, sorry, I couldn't see it. Who's raising their hand?
Son.
Oh, go for it.
Hey, Hassan, a student.
You're muted. Hassan, you're
Hassan, you're muted.
Hi, can you guys hear me?
Yeah, we can hear you.
Well, I wish you you could come to Penn in real life, but great talk. I guess I just had one quick question. So, where do you see this technology being applied in the future? Broad scope. Like, where do you think it could?
That's a good question. So like, so a lot of people who are mapping all the different cell types in the human body are starting to use CiteSeq more and more, and the more and more that we are you talking specifically about this total VI CiteSeq measurement or the lamina association stuff?
Sorry, yeah, I guess it was more of the total VI kind of correlation.
So sort of naively, like I think this is a powerful way of helping to kind of harmonize our, really well-established knowledge of, CD classification, surface markers with gene expression data, and looking at, with kind of higher resolution, different heterogeneity in different tissues. But the better way to answer that question is to ask, okay, not, which academic labs are necessarily, using this for Cell Atlas grants, but which companies are buying these antibody panels from BioLegend? And we did this project in close collaboration with BioLegend. They designed and developed this panel. And talking with them has really been fascinating for me because their biggest clients aren't people like us. They're huge pharmaceutical companies that are doing massive panels and really just trying to do much faster and higher content ways of doing facts, essentially. They're almost less interested in the gene expression analysis and much more interested in the classical surface marker identification of cells. And so some of the panels we've been privy to and some of their measurements when implementing total VI are samples where you have like different patient samples of PBMCs who have been treated with different drugs and looking for emerging cell populations that you couldn't detect with, for example, only like a surface marker or only the expression of a particular gene, but that you can see emerge when you apply all the data together into one latent space. And so, I mean, to answer your question, like I'm still learning about that, but apparently like personalized medicine and drug discovery is huge, as measured by the fact that like our connections at BioLegend care much less about like what people like us are doing with it and much more about what their bigger company clients are using.
Yeah, that's incredible. I didn't really think about the pharmaceutical application. I think one other area that is really interesting, at least to me personally, is the gut microbiome, where you have so many different populations that are so heterogeneous and so little is known about it, and there's so much impact from the DNA expression, but also the proteins that are being translated and released. So.
Yeah, and I think that there's, I don't know if it's published, but I think that there's a really cool cohort of cells of measurements on patients who've been infected with SARS-CoV-2 and looking at how, how the surfaceome, the surface proteome of lung epithelial cells is augmented in disease states. And so I think microbiome, I think disease, characterization, potentially all powerful applications.
Thank you.
Good to see you, Hassan.
Yeah, you too as well.
Yay, in my biomems class in undergrad.
Awesome. So with that, why don't we thank the speaker one more time for a fantastic talk. And I know many of you are meeting with him later today. And if you have further questions, I'm sure Aaron would be happy to receive an email, or you can reach out to him. And yeah, thank you so much, Aaron.
Great,
thank you for your attendance.
Appreciate it. I'll stay on this channel, or should I move to the next channel for the meeting, for the next round of meetings?
Kat.
Well, I might end it just so that we have time to get set up, if that's okay.
Perfect.
But if anybody wants to get in touch with you and they don't have your email, feel free to reach out to me and I can connect you guys offline.
Great. Thanks, Kat.
Yep. Thanks for coming, everybody.